Let us now consider the molecular mechanism of splicing reaction. So there are some questions. For example, how are the introns and exons distinguished? Second, how are introns removed? And third is how the exons are joined with high precision. So these are the basic questions which arise in the mind. The borders between introns and exons are marked by specific nucleotide sequences within the pre-mRNAs. So the border between exon and intron, here at this border, there are some specific sequences in the pre-mRNA. These sequences delineate where splicing will occur. So at these sequences, splicing will take place. Thus, the exon-intron boundary, that is the boundary at the 5' end of the intron, is marked by a sequence called the 5' splice sequence or splice site. The intron-exon boundary at the 3' end of the intron is marked by the 3' splice sites. So there are two types of splice sites. One is 5' splice site and the other one is 3' splice site. The 5' and 3' splice sites were sometimes referred to as the donor and acceptor sites respectively. That is, the 5' site is donor and the 3' site is acceptor. So here you can see in this figure, this is 5' side and this is 3'. And in this DNA, this is 3' splice site. And this sequence, this marks the 5' splice site. So there are specific sequences on these sites. And in addition to these sequences, there is also a specific sequence in the intron. So this is in between is intron and these are two exons. So these are the specific sequences present at the splice sites. The figure shows a third sequence necessary for splicing. This is called branch point site or branch point sequence, and this was present in the center of the intron. It is not very much in the center, but it is towards the 3' site. It is found entirely within the intron, usually close to its 3' end, and is followed by a polypyrimidine tract, that is PY tract. So here again you can see in the center it is the branch point sequence and then it is followed by PY tract. So this is important sequence. The most highly conserved sequences are the GU at the 5' site, AG at the 3' site, and A in the branch site. So these nucleotides are highly conserved. These highly conserved nucleotides sequences are all found within the intron itself. These sequences are not present in the exons because exons have to be translated into proteins. So these should not be present in the exons. This is perhaps because the sequence of most exons, in contrast to the introns, is constrained by the need to encode the specific amino acids of the protein product.